After studying this module, you shall be able to know about the significance of alcohol poisoning, classification of alcohols and importance of some notable alcohols, and the forensic importance and examination in case of alcohol poisoning. Now students, let us start with the introduction. The term alcohol is derived from an Arabic word al-kohol, which means something subtle. Alcohol causes a slowing of nerve conduction which results in slower reaction times, difficulty in processing and assimilating information. The aliphatic alcohol forms a homologous series beginning with methanol, ethanol, N-propanol, Isopropanol, etc. The first three are readily soluble in water in all proportions, but as the carbon chain length increases, water solubility decreases and octanol is almost insoluble in water. Alcohol beverages are principally a mixture of water and ethyl alcohol with small amounts of other substances which impart the characteristic odor and flavor to the beverage. These substances are referred to as congeners because they are simultaneously produced during the fermentation process. Congeners consist of organic acids and esters. Alcohol is one of the most useful alcohols for many purposes. Being a central nervous system depressant, it causes an irregularly descending type of depression. Higher centers are depressed first, followed by the midbrain and thalamus, then spinal cord and finally the medulla. The recovery occurs in a reverse order. Now we will study about the forensic issues. Alcohol is undoubtedly still the largest contributor to impaired driving, but the additional impact of drug impairment on driving has also become a focus in recent years. Forensic toxicologists play a significant role in many aspects of drunken driving, particularly in measuring the pharmacological relationship between the drug or alcohol use and impairment. They have a crucial role in analyzing these samples from motorists suspected of being impaired, interpreting results and presenting material evidence in the court. Ethanol is the most commonly encountered alcohol in terms of drunken driving cases. The consumption of ethanol is legal in many jurisdictions where it is normally considered as a socially acceptable if consumed in moderation. Other alcohols are occasionally encountered in terms of drunken driving cases. Methanol is a highly toxic substance and is not generally consumed knowingly but may be consumed by accident, particularly if the person is desperate to consume alcohol who is unaware of its toxicity. Methanol is also of major concern as it is frequently being used for adulterating liquors as a cheap substitute of ethanol in the illicit liquor manufacture syndicates, which generally results in fatal cases. Other alcohols such as isopropanol and ethylene glycol can cause impairment, but as for methanol, consumption of these substances is uncommon. Now in this table we have shown on one column the products that have been obtained from alcohol, and the next column represents the alcohol percentage by volume. Starting with the first, we have rum having the alcoholic percentage of 50 to 60%, then whiskey, gin and brandy having the alcoholic percentage by volume, that is 40 to 45%, port or sherry alcohol by volume percentage is 16 to 20%, for wine it is 10 to 15%, for beers it is 4 to 8%, and for arrack it is 40 to 50%. Now students, we will learn about the classification of alcohols. Alcohols are essentially hydroxy derivative of the aliphatic hydrocarbons. Broadly, they can be divided into three categories. Starting with the first, we have monohydroxy alcohols. This category of alcohols contain only one hydroxyl functional group. Examples of monohydroxy alcohols contain methanol or methyl alcohol, ethyl alcohol or ethanol, isopropanol or isopropyl alcohol, etc. Next category is of dihydroxy alcohols. This category of alcohols contain two hydroxyl groups and is generally referred to as glycols. And examples include ethylene glycol, propylene glycol, etc. The third category is of the trihydroxy alcohols. Trihydroxy alcohols are not true alcohols but only derivatives. Examples include propane derivatives in glycerol or glycerine. Next is some notable alcohols of forensic importance. Starting with the first, we have ethanol. Ethanol or ethyl alcohol or grain alcohol is clear, colorless liquid with a typically fruity odor and having a burning taste. It is produced mostly by synthetic protection from ethylene. This is mainly done by direct hydration process. Ethanol is soluble in water as well as lipid. The hydroxyl and the ethyl groups confer both hydrophilic and the lipophilic properties. Thus, ethanol is an amphophile. Its specific gravity is 0.79, which is 1 ml of alcohol weighing 0.79 grams. The ethanol content of various alcoholic beverages is expressed by the volume percentage or by proof, the latter being the choice of the percentage of alcohol by volume. Ethanol is toxic by oral, inhalation, subcutaneous, intravenous, intra-arterial, intraperitoneal and the dermal routes. Vaporized ethanol can be rapidly absorbed by inhalation leading to intoxication. Ethanol is a CNS depressant but produces some apparently stimulating effects initially because of depression of inhibitory control mechanisms in the brain. Several antihistaminic, decongestant, multivitamin and cough syrups contain varying percentages of alcohol from 2 to 25%. Ethanol has been popular in the past as an antiseptic. Surgical spirit used even today in mostly ethanol with a small quantity of methanol having a percentage of 90 to 95 and 5 to 10% respectively, along with the traces of castor oil and methyl salicylate. Next is methanol. Methanol or methyl alcohol is prepared by destructive distillation of wood or molasses. It burns with pale blue, non-luminous flame and its vapors produces explosive mixtures in presence of oxygen or air. Methanol, also known as methyl alcohol or wood alcohol, is a chemical compound with a chemical formula CH3OH. It is the simplest alcohol and is light, volatile, colorless, tasteless, flammable, poisonous liquid with a very faint odor. It is used as an antifreeze, solvent, fuel and as a denaturant for ethyl alcohol. Because of its poisonous properties, methanol is also used as a denaturant for alcohol or ethanol. Methanol is rapidly absorbed from the GI tract or the gastrointestinal tract through lungs and skin. Methanol itself is not toxic, but its two metabolites formed, that is formaldehyde and the formic acid, are highly toxic in nature. These compounds are responsible for causing profound metabolic acidosis and visual defect and blindness. Next in line is isopropanol. Isopropanol or isopropyl alcohol or 2-propanol is commonly known as blue heaven, as isopropanol is often highlighted blue to differentiate it from other colorless liquids, which has led to the designation blue heaven by the addicts. However, it is a colorless volatile liquid with a faint odor of acetone and a slightly bitter taste. It is generally used in massage as rubbing alcohol having a alcoholic percentage of 70%, as a disinfectant, as antifreeze, as paint remover, as cleaning solution, in toiletries like hair tonics, as an aftershave lotion, and as an industrial solvent. Isopropanol is 2 to 3 times more potent than ethanol as a CNS depressant, and the usual fatal dose of isopropanol is about 250 to 300 milliliters. Isopropanol can be absorbed through all routes. In the body, it is rapidly metabolized by alcohol dehydrogenase. Approximately 80% is converted to acetone and the residue is excreted unchanged in the urine. Acetone is excreted in the urine and breath and also metabolized to acetate, formate and carbon dioxide. Isopropanol may be generated spontaneously in a dead body, presumably due to the bacterial or other putrefaction processes. This fact must be borne in mind when subjecting viscera to chemical analysis. Next is ethylene glycol. Ethylene is also known as 1,2-ethanediol or glycol alcohol. It is a colorless, thick, odorless, non-volatile liquid with a bittersweet taste. Ethylene glycol is not absorbed through skin and because of its low vapor pressure does not produce toxicity upon inhalation. It is however rapidly absorbed through the gastrointestinal tract and metabolized more than 80% to glycoaldehyde, glycolic acid and oxalic acid, which inhibits the diverse metabolic pathways in the body, including the oxidative phosphorylation. Other metabolites include glyoxylic acid, glyoxyl, formic acid, glycine, oxalamate, malate, benzoic acid and hippuric acid. The usual fatal dose of ethylene glycol is about 70 to 100 milliliters. Next, we will study about the metabolism of alcohols. Alcohol is readily absorbed through the stomach and in particular through the small intestine, with effectively 100% of an oral dose absorbed in a healthy person. It transfers readily to the bloodstream owing to its high water solubility and is then distributed rapidly throughout the body, distributing into all the tissues, particularly those having a higher water content. Alcohol is metabolized in the liver, although some is eliminated unchanged. Although the major pathway for metabolism is oxidation via acetaldehyde to acetic acid, a small percentage of ethanol is conjugated to give ethyl glucuronide and ethyl sulfate. About 90% of the alcohol absorbed is oxidized in liver and rest 10% is excreted. Alcohol is oxidized to acetaldehyde by alcohol dehydrogenase, that is ADH, and coenzyme nicotinamide adenine dinucleotide, that is NAD, in the liver. This acetaldehyde is converted into free acetic acid or acetyl coenzyme A. The acetate undergoes oxidation to carbon dioxide and water in the citric acid or Krebs cycle. This acetate can form glycogen, proteins, fats, cholesterol. Being volatile, alcohol can diffuse into free air and hence can pass from the pulmonary system into the lungs, where it is expelled in breath. This excretion of alcohol in breath makes breath alcohol testing possible. Food present in the stomach can slow down the absorption rate of ethanol. Next, we will study about the mechanism of alcohol. Generally, ethanol is a central nervous system depressant. The precise mechanism by which the alcohol affects cognition and psychomotor control is not understood completely. Alcohol produces simultaneous changes in many neurotransmitters and increases the fluidity of neural cell membranes. Alcohol in small doses interferes with the cortical function like conduct, judgment, self-criticism and release of the inhibitory tone, but in larger doses depresses the medullary processes. Alcohol intoxication is also called as acute alcohol poisoning or inebriation. Next, about the forensic examination. Starting with first, we have ethanol or ethyl alcohol. For the detection of ethanol, various tests are to be carried out in the exhibit. One of them is the iodoform test. Appropriate amount of the sample is taken and about 1 milliliter of 5% sodium hydroxide solution is added to it, and subsequently the iodine solution is added drop-wise with shaking until the liquid becomes persistent dark brown in color. The whole is left for few minutes. If the iodine color disappears, more drops of iodine solution are added drop-wise until persistent brown color of iodine reappears. Few drops of dilute sodium hydroxide solution are added to remove the extra iodine. Equal volume of water is added and left for about 10 minutes. Observation of yellow crystalline precipitate indicates the positive test for the presence of ethanol. Next is the dichromate test. Adequate amount of sample is taken and about 0.2 milliliters of 2% potassium dichromate solution is added, followed by about 1 milliliter of concentrated sulfuric acid. The yellow color of the dichromate changes to green or blue, indicates the presence of ethanol. Next is sulphomolybdic test. The sulphomolybdic test is actually a test having 1 gram of molybdic acid in 25 milliliters of concentrated sulfuric acid. Now for the detection, 2 milliliters of the hot reagent is added to 2 milliliters of the distillate. A deep blue ring appears at once at the junction of two liquids. On shaking the whole mixture, it becomes deep blue. It indicates the presence of ethyl alcohol. This test is highly sensitive and is negative with acetone, acetaldehyde and even dilute solutions of methyl alcohol. A strong solution of methyl alcohol gives only a light blue color after several minutes. Next are the test for methanol or methyl alcohol. For the detection of methanol, various tests are to be carried out. Starting with the first, we have the chromotropic acid test. Appropriate amount of sample is taken in a test tube and about 2 milliliters of potassium permanganate solution is added to it and shaken well. After that, few crystals of sodium bisulfite is added with shaking till the disappearance of the color of the solution. About 1 milliliter of the chromotropic acid, that is 5% of aqueous solution of sodium salt of chromotropic acid, and subsequently concentrated sulfuric acid is added slowly with inner side walls of the test tube. to the extent of 15 milliliters. Appearance of violet color indicates the presence of methanol. Next test is the Schiff's reagent test. About 4.5 ml of sample is taken in a test tube and 0.5 milliliters of ethanol is taken. 2 milliliters of 3% potassium permanganate solution and 2 ml of phosphoric acid is added to it and left for 10 minutes. After that, 1 milliliter of 10% oxalic acid is added, followed by 1 milliliter of concentrated sulfuric acid. The contents are left to cool at room temperature. 5 milliliters of Schiff's reagent is added further and kept for half an hour. Appearance of purple color indicates the positive test for presence of methanol. Next, we'll come to isopropanol or isopropyl alcohol. The acid distillation of the exhibit is used to separate the isopropyl alcohol from tissues and other biological material. The distillate is subjected to the test that is I am going to tell you about. 2 milliliters of distillate is taken in each of the 2 test tubes and 3 drops of potassium permanganate in phosphoric acid by dissolving 3 grams of potassium permanganate and 15 milliliters of syrupy phosphoric acid in 85 milliliters of water is added in one of the test tube, and this is allowed to stand for 5 minutes. The color, if any, left after oxidation is decolorized with a pinch of sodium bisulfite. After that, 1 milliliter of 10% sodium hydroxide and 1 milliliter of 5% furfuryl are added to both the test tubes. The contents of each of the test tubes are filtered into test tubes containing 2 milliliters of the concentrated hydrochloric acid. A pink ring is formed at the junction indicating the presence of isopropanol, and the pink color in the other test tube indicates the presence of acetone. Now students, I will summarize all that we have studied in this molecule. Alcohols are hydroxy, that is monohydroxy, dihydroxy and trihydroxy derivatives of the aliphatic hydrocarbons. Generally, the polyhydroxy alcohols does not exist as the hydrogen ion of an hydroxyl group conjugate with the other hydroxyl group to create the water molecule. Thus, they form aldehydes, ketones and carboxylic acids. Absorption of alcohol is slower in the stomach than the intestines, where it is faster in empty stomach than when it contains food. Food affects the absorption rate of alcohols by delaying the emptying time of the stomach. Fatty foods, proteins and starch delay the absorption rate. Milk has also similar effect. Food prevents 10 to 20% of the ingested alcohol from being absorbed. Diluted form of alcohol is absorbed slowly than the concentrated form. The type of beverage also affects the rate of absorption. Whiskey is absorbed rapidly and its complete absorption occurs within 1 to 3 hours. The absorption of alcohol depends on habituation and tolerance of the individual, as habituated heavy drinkers tend to absorb alcohol more quickly. Section 85 of the Indian Penal Code says that nothing is an offence which is done by a person who at the time of doing it is by reason of intoxication incapable of knowing the nature of his act, or that he is doing what is either wrong or contrary to law, provided that the thing which intoxicated him was administered to him without his knowledge or against his will. Section 85 of the Indian Penal Code gives the same protection as Section 84 does to a person of sound mind who is by reason of intoxication is incapable of knowing the nature of the act, or that he is doing what is either wrong or contrary to law, provided the thing was given against the will and without the knowledge. Under section 86 of the Indian Penal Code, a person who gets into a state of intoxication voluntarily presumed to have the same knowledge as he would have had he not been intoxicated. Alcohol abuse is prevalent worldwide. Efforts have been made to totally prohibit alcohol consumption in many countries without much success. However, such attempts usually lead to illicit brewing and consumption with even more serious consequences. Proof spirit refers to a standard mixture of alcohol and water of relative density, that is 12 by 13 at 51 degrees Fahrenheit, that is 49.28% of alcohol by weight. Proof strength of alcoholic beverages is expressed in degrees.